Jai Hind students. So let's start with the new topic and the new chapter of biology. So today we are going to start with the chapter number 8, which is cell, the unit of life. So now let's begin with the chapter. As you know that each and every living body is made up of cell, right? So if you will see about the organisms, you will come to know that there are some organisms who are unicellular, whereas some are there who are multicellular, right? For example, if you look out for the unicellular organisms, a very common example is there of amoeba. Whereas for multicellular, so many examples are there like the lion, human, mango tree. So many are there which are the examples for the multicellular organisms. So now let's see about the different types of cells which are present in our environment or which you can see or study. On the basis of types of cells, there are two different divisions for this. One is prokaryotic cell, whereas the other one is eukaryotic cell. So let's first study about the prokaryotes. Before knowing any other thing, let's see that what are the prokaryotes, because very first thing comes in the mind that what is prokaryote actually. So prokaryotes are all the moneras. In your previous chapter, you have already gone through the moneras. In monera class, all the bacteria come. So whatever bacteria you see, they all are from the monera kingdom, right? So all the bacteria comes in the kingdom monera and they all are prokaryotes. Now why they all are prokaryotes? Because the very first thing, they have no well-defined nucleus. So no well-defined nucleus. Not only this, in fact, just because they don't have a well-defined nucleus, they are not having the other Golgi bodies. Now, what are these Golgi bodies? Golgi bodies means your mitochondria, endoplasmic reticulum, rough endoplasmic reticulum. These all are the Golgi bodies, right? So the moneras or the prokaryotes, they don't have Golgi bodies, but only one is very commonly present in the prokaryotes and the eukaryotes both, and that is ribosome. Wait. This question is often asked in exams, that which one is there which is present in both eukaryotes and prokaryotes both. So you're going to answer that that is the ribosome which is present in the prokaryotes and eukaryotes as well. Okay, so these are some characteristic features of the prokaryotes which you can see in the moneras or the bacteria. Now let us define eukaryotes first, or let us understand this first. So as example, we have discussed eukaryotes are having quite well developed body. For example, you can say the plant, you can say animals. Alright. So the very first thing about them eukaryotes is that these eukaryotes are having well-defined nucleus. So they have well-defined nucleus. They are having Golgi bodies because we need energy, and you know that because of mitochondria we are having energy, right? So Golgi bodies are present. Third, yes, they have ribosome. So they are also having ribosome. And this is very important question asked in generally in competitive exams. Okay, so this is the difference between the prokaryotic cell and the eukaryotic cell. So now let us move ahead and let us see first new things. So now let's see what was the cell theory actually. There were the two scientists called Shevan and the other scientist who was Shelvin, and he was botanist, whereas Shevan was a zoologist. They have done the experiment, and because of their experiment they come to know some closure distributions, or they come to know about the cell differentiation and all. So they have given the cell theory. Okay, so they gave some cell theories, they differentiated that how plant cells are made and all. But later on, you know that about the cell, who has discovered the cell is a Robert Hooke, right? So he has discovered the cell, but later they have also practiced on the plants, they have practiced in the animal cell together. But later on there was another scientist whose name was Redolph. Now this Mr. Redolph, he has gone through the experiments and has modified. So he modified cell theory. According to the Redolph, he said that all the organisms are made up of cell and its product. So he said all organisms are made of cell and its product. Then he also said that all the organisms are created from the pre-existing organisms. So organisms are made or created cause of pre-existing cell. Maybe you in this line you are having any doubt. So all the organisms are made from the pre-existing cell because they all are the daughter cells, right? So if a plant is there, then from the plant seed the another plant is going to develop, right? And it's going to have because of the gametes of the particular plant, right? So from one cellular body the other cellular body is developing, and that is how the continuation of the species are going on. So Redolph has modified the cell theory and has given these different about the modifications, about the result of the cell, about the classifications of the self, right? So just the same way as Shevan was there, he has also said that the plants are having the plasma membrane. Okay, so as Redolph, he also said that plants are having something like this called the plasma. Animals are having the plasma membrane like this. So this was given by them. Now let us see that about the shapes of, about the sizes of different cells. So the very smallest cell which you can see is of the PPLO. So the smallest cell is of PPLO, which is a bacteria. Okay, so PPLO is a type of a bacteria which is also you can say of the mycoplasma. Mycoplasm. Okay, then there is the largest cell. Largest cell which you can see is the egg of ostrich. Okay, so ostrich egg. Then we are having the longest cell. Longest cell, if you will talk about, then you are going to say about the neurons or the nerve cells. So the nerve cell is the longest cell present in our body. Fine. So now on the basis of all these are the actually lengths of the cells, right? So these are the sizes. Now let us categorize the types of monera which you can see. Types of monera, that is your bacterias. So on the basis of their shape, they have been classified in 4 different types, right? So if you see the circular one, right? So it is the bacillus. If you remember the previous chapters, you will recall yourself, then the circular one are the bacillus bacterias. If you see, sorry, somewhat oval shaped. Okay, like this. And if you see the circular, these circulars are cocci, right? If you see comma shaped, these are the vibrios. Remember? And if you see somewhat zigzag like this, these are the what? Can you guess? Okay, so these are the spirilla. So on the basis of their shape, they have been differentiated into 4 different types, right? Now their outer covering can be of different types also. Okay, so these prokaryotes, these moneras, if you will see, you will find that they are having 70S type of ribosome. Further we will see that what it is. Okay, so they are having 70S type of a ribosome, whereas in the eukaryotes, if you will see, they are having 80S type of a ribosome. This is actually a type of ribosome which you can see. Fine. So now let us see that on the basis of this, what are the different, apart from their shape. On the basis of shape they have been classified, right? Apart from this, there are 2 more other types of bacteria present on the basis of their nature. So on the basis of the nature, you can see 2 more different types of bacteria. For example, gram positive and gram negative. The word gram is here just because of the scientist, because the scientist who has discovered this name was Gram. Alright. So the gram positive bacteria are those bacteria which can be stained, right? So they can got through the stains. If you are staining them, they can be easily stained. So these staining bacteria are called as the gram positive. Whereas the gram negative bacteria are those bacteria which can't be stained, right? So these bacteria cannot be stained. That is why they are called as a gram negative, and these can be stained, so they are called as the gram positive bacteria. Now let us see further that what is the different outer cellular structure of the bacteria, right? So on the basis of how they do, their coverings are there, how their coats are there, let us see that now. So let's see about the prokaryotes, what are the different cellular body they are having. Okay, so the bacteria are having three more different outer bodies, like they are having flagella, flagellas are there which helps the bacteria to do locomotion. A bacteria may be single flagellated, may be double flagellated, or may be the multiflagellated. Later on we'll see the structures of them, but let us see first this now. So flagellas, they help in the locomotion of the bacteria. The next we are having pili. This body part in the bacteria, it is there for the sexual reproduction. So this help in the process of reproduction in the bacteria. And then there is a fimbriae. So fimbriae is there in the bacteria found to help them to get attached with the substratum. Attachment with substratum. Okay, so this one is meant for attachment of the substratum in the bacteria. So fimbriae is here really very important for you to know. So now this is about the different parts which are present in the bacteria. Now let us see some extra about the structure of the bacteria. Few more things are there. Now let us see that first. Alright. Structure of the cell, right? So here you can see a structure of a typical cell, bacterial cell. As here this you can see the outer body is there, right? Now on this covering inside the body you will see undifferentiated nucleus, right? So that undifferentiated nucleus in the bacteria are called as nucleoid. This is having all the genetic material, the genome in it, which is going to be transferred to the next generation. Now next over here you can see this cellular structure. This circular structure is extra chromosomal circular DNA, which you can also call as a plasmid, and this DNA is a double stranded. Okay, so this one is the extracellular chromosomal DNA, which is also a plasmid. Now the work for this is to resistance to the antibodies. It helps to resist for the antibodies. Now the outer covering you can see is the cell membrane, and this cell membrane is there for its the protection. Okay. Now the bacterial cell is also having a mesosome, which is there to give the energy to produce ATP, because they also need to do different life processes, life activities. So for that, as we are getting the energy from mitochondria, they are getting it from the mesosomes. Now the something is present in the bacteria which gives them the color, and that part is called as a chromatophore. So chromatophore is there in the bacteria which give them the color. Now let us see the protective unit of bacteria. Here you can see the three different layers, and these three different layers are the protective units of the bacteria. The outermost which you can see is the glycolysis. This middle one inner you can see as the PM, which is the plasma membrane, and then the last you can see the cell wall. Okay, So the cell wall is the inner one and this PM is the middle one, which is there as a protective unit in the bacteria. So hope everything is clear to you about the prokaryotes. Thank you for watching the video.